This is not an assignment, but it's definitely worth doing. My students last semester said that this was one of their favorite things that they helped them learn the most. So if you're struggling with the levels of structure with proteins, give this thing a go. All you need are some pipe cleaners and various amounts of pony beads. You just need 2 different colors. All right, so what we're gonna do is I'm gonna use my lovely random number generator. It looks like a 20-sided dice because I am a nerd. These are most of the dice that I have kicking around my office, not all of them. All right, so what I'm gonna do is I'm gonna have a color of pony beads that are going to represent my hydrophobic amino acids. I'm going to choose that as yellow because yellow seems like a scaredy color to me. And then my hydrophilic colors are going to be this blue color, blue because water's blue. I don't know, use your own logic. That's what I decided. Okay. And in these instructions, I said to roll the dice, and if it is an odd number, then we're going to add a hydrophobic, and so that was, I rolled a number 17. So I'm going to add my first amino acid on my protein chain is going to be a hydrophobic amino acid. Okay, and right now I'm just going to build the primary structure of my protein. Okay, and I'm just going to roll this. I'm going to get about 10 amino acids on this. I'm going to keep them about an inch apart, which is about the width of my thumb. That's how I'm measuring. All right, so that one was hydrophilic, so I'm going to add a blue. And just a pro tip, I actually took 2 pipe cleaners and twisted these together. That way the pony beads don't slide around so much. Kind of keeps them a little bit, keeps them where I put them a little bit better. I'm happy with that amount of amino acids. To prevent you from having to sit here and watch me do this a whole bunch more times, I went ahead and made another one that's really long. Okay, so what I'm going to do now is twist these 2 together. And so now I have this one really long polypeptide chain. And at the moment, all I have is the primary sequence of just the order of my amino acids. So now what I'm going to try to do is to set up the secondary structure where I'm going to fold my protein. I'm trying to bunch up my hydrophobic amino acids so that they're interacting with each other, and having my hydrophilic amino acids sticking out into what would be my watery substrate. And I'm kind of beta folding this. Once we get this kind of set up with our secondary structure, then we can mess around with getting our 3-dimensional structure set up. Here's my 2-dimensional structure. Now what I'm going to do is try to fold this into a 3-dimensional structure, and now I really need to make sure that all of my hydrophobic amino acids, those are my yellow ones, are going to be kind of shoved in the middle. And I really want to have my hydrophilic, so the blue ones in this case, amino acids, be surrounding those. So this can be really kind of tricky if you have an extra set of hands that can help you hold hold things while you shove. Oh, and this amino acid's about to fall off, so I'm gonna encourage him to stay. And that amino acid really needs to get in there. Okay. All right, now I just have this one little dude. He's gonna go like that. And there is an estimate of the three-dimensional structure. All my yellow amino acids that are the hydrophobic are on the inside, and all my hydrophilic are on the outside, interacting with the water. And that's just, this is an example of the tertiary structure. Now, what I would usually do in my in-person classes is that each group has made their own. They all come together with their their polypeptides, and then together we make a protein in the quaternary structure. It's kind of a fun little activity. Give it a try, see if that helps you kind of visualize that a little bit better.